Hello everyone, this is Mr. Jackson, and today we're going to be continuing Unit 3 by going into Lesson 2. In Lesson 1, we discussed cell growth and the need for cell division so that cells do not become too large. And today we're going to be looking into the actual process of cell division. As far as our learning objectives go for this lesson, you'll know that you've mastered what you need to. Whenever you can, explain the role of chromosomes during this cell division process, describing the main events of the cell cycle, and describing what happens during the phases of mitosis. Okay, so we're going to be looking at the specific roles of chromosomes and why they're important to this cell division process. We're going to be looking at the cell cycle, and within that cycle, we're going to be looking at the individual phases of what's called mitosis. So the first thing to point out whenever we're discussing why chromosomes are so important to us is we're going to want to point out that, cells have a tremendous amount of genetic information in the form of DNA. Okay. Now, DNA represents all of the information, the blueprint for how the cell makes literally all of the proteins that are required in order to build cell organelles and all of the different molecules required for cell to function. Now, this is a tremendous amount of info. An easy example for what this might be is like if you look at the bacterium E. coli. If I were to stretch all of the DNA in an E. coli bacteria, it would stretch and measure at about 1.6 millimeters. Now, while that doesn't sound very long to us, it is actually up to 1,000 times longer than what the cell would actually be. So that would be like if I handed you a 300-meter rope, right? So about the length of 3 full football fields. And I said, could you please fit this down into your backpack? So naturally, we need to have a better way of handling such large DNA molecules, so much information. And we do that by carefully bundling all that info into packages that we call chromosomes. So I've shown you here in prokaryotic or more simple organisms, in those cells, DNA is packaged into a single circular chromosome. There's not a whole lot of information there because those cells are a lot more simple. Whereas in eukaryotic cells, for multicellular organisms such as ourselves, DNA has to be packaged into multiple chromosomes. As these cells are a lot more complex, they require more information, and it probably wouldn't be the most efficient thing to pack that much info all into one. And so a good example is in fruit flies. They'll have 8 chromosomes carrying all of their genetic info. Carrots would have 18. So carrots are actually more complex, requiring more chromosomes than a fruit fly. And then humans, we actually have 23 pairs for a total of 46 chromosomes. We're going to have more than enough time to explore that in later lessons. But why do cells go to such lengths to package their DNA into these chromosomes, into these packages? And the main reason is that it ensures that we can equally divide our DNA so that both cells at the end of cell division, whenever we go from one cell to having 2 genetically identical cells, we want to have the full genetic material separated equally into both of those so that each of those cells can then function independently. So chromosomes are going to be our way of precisely separating into 2 daughter cells during the process of cell division. And I did want to break down these levels of organization. I think that this is a very, very useful way to look at the overall structure of DNA and how that will actually build to the overall structure of chromosomes. We are actually going to be looking at DNA structure in a lot more detail in a future lesson, and you'll even have the opportunity to have some hands-on building where we'll actually build DNA molecules by hand and see how those that structure can actually be used to package and condense all of that information. So if you look here on the right side, you see a DNA, what we call a double helix. So this is just basically 2 strands, a left and right side, and it typically is going to look like a twisted ladder. So it's kind of like I had rungs of a ladder and then I twisted it to create a coil. Now, this DNA, we need to keep it neat and condensed. And so what we're going to find is that DNA is then going to coil and wrap around these little proteins called histone proteins. And whenever I have a little bead-like structure, a little bundle of DNA wrapped around these proteins, I call that a nucleosome. Whenever I have a bunch of nucleosomes, they can actually come together and tightly compact into coils. You take enough of those, you get supercoils. And then if you compact all those supercoils and you see them all together, that is what we're actually looking at whenever we're seeing a chromosome. Now, importantly, this X shape is actually two sister chromatids. This is what a duplicated chromosome looks like. So typically, there would only be a single line, a single strand, and that would be a chromosome. But in this case, we can see that we have already duplicated, we've already made a full copy of all the information in this chromosome. And so we have one side of that chromosome here, and the other duplicate identical copy here. And we'll also cover this in greater detail in more lessons to come, but the little part right here that joins those two sister chromatids, those copies of each other, we call that a centromere. It's in the center. So we're going to specifically focus on eukaryotic, multicellular organisms and the way that their cells divide, because that applies directly to us. And so it's a really good way of coming to learn these steps. Now, cell cycle, whenever I refer to the cell cycle, what we're actually talking about is a series of 4 repeatable phases. And the length of each phase, meaning the length of the entire cell cycle, is going to vary depending on what type of cell we're talking about. For example, a red blood cell only completes its cell cycle in 4 months, right? So give or take about one full life cycle of a red blood cell being 4 months, whereas a lot of other types of cells will last much longer. And we want to go ahead and address one key term here. We call this the in-between period of where a cell is actually doing the majority of its growth before it's going to divide. And we call that little in-between phase interphase, right? So very, very similar. So interphase, think in between. And so we want to break down what's actually happening during interphase. It's a series of 3 steps. We have the first phase, which is called the gap 1 or G1. This is where the majority of cell growth is happening. So all of that growth that, ultimately we want to limit. We don't want to get too big, as we discovered in lesson 1. But this is where the cell's going to increase in size. We're making a bunch of new proteins and all the organelles that'll be required in the new daughter cells. After gap 1, we go into what's called S phase, and that just stands for synthesis. This is where we're creating something new. We're synthesizing. And what we're actually making in this case is a full copy of our DNA. So we're seeing DNA replication. So basically, new DNA, we synthesize it. The chromosomes are completely copied or replicated. And this is going to result in a full copy of all the genetic material. So now we can actually divide 2 full copies one into each new daughter cell. And then finally, the gap 2 phase, G2, this is where we're actually preparing for cell division. So any final organelles or molecules that might be required for cell division to happen, we're going to go ahead and make sure that we produce those as well. Might have a little bit more growth of the cell, but again, most of that happened during G1. And then we get to M phase. This is our cell division phase. Now, M phase might be useful to remember cell division. M could stand for mitosis, but it is important to understand that M phase is not strictly mitosis. It's actually 2 parts to complete the actual division of the cell into 2 daughter cells. So we are creating those identical daughter cells from one parent. And there are 2 main stages. We're going to explore mitosis in each of its 4 phases in more detail. But whenever you think of mitosis, remember that that's only dividing the cell's nucleus. This is going to specifically focus on all of the copied genetic material that we have making its way into 2 nuclei rather than just 1. And an easy way to remember these 4 phases is just by remembering the acronym PMAP. And that'll be the order in which these phases occur. And then finally, we also have the process of cytokinesis. This is that last step in the cell division process where we're finishing dividing the last of a cell's cytoplasm. This is where one cell becomes 2. So first phase of mitosis is called prophase. Now this is going to be where genetic material inside the nucleus, we're going to condense it and we're going to be able to visibly see it in the form of chromosomes. And once that's the case and we have it nice, neat, and condensed, we're actually able then to make those copies. And we can see those 2 sister chromatids for each of these chromosomes inside the nucleus. Outside the nucleus, something else that's very important is happening where we actually have what's called a spindle or spindle fibers start to form. We have these centrioles, which are very, very important for cell division. They're going to make their way to 2 separate sides or poles of the cell itself. And ultimately, this is going to be the direction in which these chromosomes are pulled into 2 new cells. After prophase, we're going to go into phase 2, which is called metaphase. This, it starts with the letter M. Easy way to remember this phase and distinguish it by a visual here is that this is where we're going to basically see all of our genetic material lined up in the middle. So you think meta, think middle. This is basically where the centromeres of all the duplicated chromosomes, they're lining up in the center, so in the middle of the cell. Spindle fibers that we addressed in the previous phase, those are now going to connect to those little center points for each of our chromosomes. And what those are going to do is ultimately we're going to then create those connections, and hopefully those identical copies, each chromosome will have one sister chromatid pulled to one side of the cell and the identical copy of each chromosome pulled to the other side of the cell. And then we get to phase 3, which is called anaphase. You see that anaphase starts with the letter A, so does the word away or apart. Really easy way to remember this phase. So once they've lined up in the middle, now what we're seeing is that we've actually, we're pulling each sister chromatid into its own side of the cell. This will ultimately be, once we have these chromosomes in each side, this will ultimately be the, the separate information for the 2 identical cells. And so we can now basically call those sister chromatids, these are now individual chromosomes, they're their own thing, and they have all the information we need for the 2 new cells. So these chromosomes are separating, they move along those spindle fibers towards the opposite ends of the cell, towards those centrioles. So you think anaphase, think they're moving away from each other. And final phase of mitosis, we've got telophase. This is where the chromosomes, they were originally distinct and condensed. Now what they're going to do is they'll basically just spread out into a tangle that we call chromatin. Chromatin is just a fancy term for when we have DNA coiled around those histone proteins. And then basically now that we have all of that information in 2 separate places, what you're going to see is 2 new nuclear envelopes, which will be the outer boundaries of those nuclei. Those are going to start to form around each little cluster or grouping of chromosomes that are, of course, now in 2 separate places. And so you can really start to see here in telophase how we're just about there. We're almost to 2 fully developed, fully containing all the genetic material that they need in those 2 areas, and they'll become 2 different distinct cells. And so now we'll complete our process with a phase called cytokinesis. This will complete the process of cell division by dividing what was then 1 cell with twice as much info as needed into 2 cells with exactly the information they need. So basically, cytokinesis is just that final parting of cytoplasm, maybe a few cell organelles, into 2 different parts, 2 different cells distinctly. And finally, we're going to end this conversation with a quick review of the cell cycle process. Up at the top here, we have the in-between phase of cell division. We called that interphase. Remember, we've broke that down into the gap 1, where we were doing the primary amount of cell growth, the S phase, which was all about making an identical copy of our DNA, and then the gap 2, which is like those final preparations before we start dividing. Whenever we start the process of cell division, we have begun mitosis, and so we broke down those 4 phases as well. The first phase was prophase. Remember, easy way to remember the order of these, PMAP. So prophase, again, this is where we see that condensing of genetic material, and we see that nuclear envelope start to dissolve, some spindle fibers, which will be that line of pull. We see those as well. After prophase, we go right into metaphase. Hopefully you remembered that. Metaphase, think M, middle. This is where we see all of those chromosomes, those copies, basically condense in the middle of the cell. Next phase is anaphase. And so this is where we remember that it starts with the letter A. This is where they move away or apart from each other. One copy will go towards one end of the cell, another copy will go towards the other, and will completely separate or move away. And then finally, for mitosis, we have the phase of telophase. Telophase is where now we have all of the information we need into 2 distinct regions of this cell, and so we can actually now build a nucleus around each of those separate groupings of chromosomes. And then we're going to finish the process of cell division here with cytokinesis, final division of, let's say, cytoplasm into 2 distinct areas. And then once we have that division occur here at the center where was previously 1 cell, we are now going to cleave this off, and we'll have 2 genetically identical daughter cells. And so that's going to conclude our conversation today over the process of cell division. Very important to understand the process, because as we move forward, we're going to want to understand how we regulate that cell cycle, how we differentiate the types of cells that we can make in that cell cycle. And so if you think back to those I CAN statements, we want to understand the importance of chromosomes. We want to be able to describe, or at least identify, the steps or the phases in cell division. And then more specifically, within cell division, we want to be able to identify the phases of mitosis, right? So all of those come together to give us a really good sense of how we can go from 1 parent cell to 2 genetically identical daughter cells. And so that'll conclude our lesson 2 lecture, and I hope that you found this helpful.